Welcome back to the deep dive. Today we are entering the ICU, and I want you to picture a scenario that is keeping doctors awake at night. You have a patient with severe pneumonia, but it's not just any pneumonia, it's a superbug.
Right. Specifically, carbapenem-resistant gram-negative bacteria. A real mouthful.
The lungs are filling with fluid, the patient is essentially drowning, all the standard antibiotics are just bouncing off.
It's a complete nightmare scenario, and it's becoming, more and more common. When you get to this point, the medicine cabinet is pretty much empty.
Well, not completely empty. There's one drug left on the shelf, isn't there? Polymyxin B.
Ah, yes, Polymyxin B or PMB.
Yeah.
In the medical world, we sort of think of PMB as a toxic sledgehammer.
A sledgehammer?
It's effective. I mean, it kills bacteria.
Yeah.
But it comes with this massive cost. It's notorious for nephrotoxicity. It destroys the kidneys and neurotoxicity.
So you're stuck. You have a choice. Let the infection kill the lungs, or use the drug and potentially kill the kidneys.
Precisely. And there's a second layer to this whole tragedy. Even if you are willing to risk the kidneys, PMB is just chemically terrible at getting into lung tissue.
You can't get to the battlefield.
Exactly. It binds to other tissues poorly, and it just can't cross that blood-air barrier efficiently. So you're flooding the entire body with a toxin, but almost none of it is actually reaching the infection.
Which brings us to the paper we are diving into today. It's from Nature Communications. And the researchers decided that if the drug can't get to the lungs on its own, well, it needs a vehicle.
It needs a ride.
And the vehicle they build. Honestly, I had to read the methods section 3 times. Are you telling me they're curing pneumonia using lemons and fake bacteria?
It sounds like a kitchen experiment gone wrong, doesn't it?
It really does.
But the paper, neutrophil-mediated delivery of hybrid cross-species nanovesicles, is actually a masterclass in bioengineering. They have created a Trojan horse, and the technical name is PMB LNV-SyBV.
That is a mouthful. Okay, so our mission today is to decode that alphabet soup. We are going to explain how you turn a lemon into medicine, how you build a ghost bacteria, and how you trick the immune system into being a taxi service.
And we should look at it exactly like a manufacturing process. There are 2 raw ingredients. So ingredient A is the lemon.
This is the LNV part of the name lemon-derived nanovesicles. Are we talking about just, squeezing juice into a test tube?
It starts with juice from citrus limonelle, but the processing is really intense. They use ultracentrifugation, spinning the liquid at just incredibly high speeds to isolate these tiny, tiny particles.
What are they?
They're double membrane vesicles, about 140 nanometers wide.
So tiny fat bubbles from the fruit. But why lemon? Why not an orange or a grapefruit?
It all comes down to the chemical composition. They analyze the lipids, the fats that make up the bubble wall. And these lemon vesicles are incredibly stable. They're made of about 44% sphingolipids and almost 40% glycerophospholipids.
And sphingolipids, those are the structural heavyweights in the cell world, right? They make things tough.
Exactly. They make the vesicle tough. But the researchers didn't just pick lemon for its shell. They picked it for what's inside. These vesicles naturally contain active metabolites, things like limonene, asparagine, and flavonoids like rutin.
And those are more than just vitamins.
Oh, much more. They're functional. They are potent anti-inflammatories. You have to remember, in pneumonia, it's often the body's own immune response, that cytokine storm, that kills the patient by flooding the lungs.
Right.
So the researchers actually compared lemon vesicles against ginger and grapefruit vesicles.
I saw that in the paper. The ginger did not go so well.
Not at all. The ginger-derived vesicles were actually toxic to the cells at higher concentrations. The grapefruit ones were safe, but they didn't really do anything for inflammation. The lemon vesicles were the Goldilocks solution.
Just right.
Just right. They suppressed pro-inflammatory cytokines like IL-6 and TNF-alpha. So even before we add the antibiotic, the wrapper itself is a form of medicine.
Okay, so we have a safe anti-inflammatory lemon bubble. That's component 1. Now for component 2, which is the one that sounds frankly dangerous, the SyBV, or synthetic bacterial vesicle.
This is where the Trojan horse part really comes in. They start with Escherichia coli, E. coli bacteria.
But wait, you can't inject E. coli into a patient. That causes sepsis.
Correct. So they have to detox it. They basically turn the bacteria into a ghost. It's a 3-step renovation.
Okay.
First, they use an enzyme, lysozyme, to eat away the outer layer. Second, they use sonication high-frequency sound waves to break the membrane open.
And then comes the chemical wash. The paper mentions something called sarcosyl.
Yes, the sarcosyl wash at a high pH. This is the most critical step. Imagine stripping a house down to its studs. This detergent just washes away the inner membrane, the DNA, the RNA, and most importantly, the toxins.
You mean the LPS?
Lipopolysaccharide, yes.
Yep.
That's the molecule on bacteria that triggers septic shock. This washing process reduces the LPS content by 19 times compared to a natural bacterial vesicle. It's a huge drop.
But hang on, if they strip everything away, doesn't it just become invisible? The whole point is that we want the body to see it.
And that is the absolute genius of it. They wash away the danger, but they keep the ID card.
Ah.
The proteomic analysis showed that these synthetic shells still have their outer membrane proteins, specifically OMP, OMI-C, and OMPIF.
Those are the flags that the immune system looks for.
Exactly. So the SyBV is decoy. To your immune system, it looks like a threat, but it's actually hollowed out and way, way safer.
Hey, we're about 5 minutes in, so let's just pause and do a quick recap. We've got some complex pieces on the table here.
Let's lock it in.
So the problem, we need to get a really toxic antibiotic into the lungs without killing the kidneys.
Right.
Component 1, the lemon nanovesicle, or LNV. It's a stable, lipid-rich shell that also happens to fight inflammation on its own.
And component 2, the synthetic bacterial vesicle, or SABV. It's a hollowed-out E. coli shell. We've stripped out the toxins, the LPS, but kept the surface protein so the immune system can still spot it.
So we have a lemon bubble and a bacterial ghost. Now, part 2. Construction. We have to fuse these two things together and load the weapon. The paper says they needed a glue to get them to merge.
Right. If you just mix lemon lipids and bacterial skins, they don't naturally want to become one single thing. They needed a stabilizer. And they added exogenous cholesterol.
Cholesterol usually gets a bad rap, but here it's playing a structural role.
In membrane biology, cholesterol is like the mortar in a brick wall, and bacteria, they don't naturally have it. By adding about 5% cholesterol to the mix, they thickened the lipid bilayer. And this had a huge side benefit. It massively increased the drug loading capacity.
How much of a difference are we talking?
It's night and day. Without cholesterol, they could only get about 1.2% of the antibiotic inside. With cholesterol, that jumped to 11%.
Wow, that's a 9-fold
A
increase.
huge increase in the payload.
Okay, that brings us to the part that really confused me at first. How do you actually get the antibiotic inside? Polymyxin B is a molecule. You can't just push it into the middle of a sphere.
This is one of the coolest physics tricks in all of drug delivery.
Yeah.
It's called remote loading, using a pH gradient.
Okay, walk me through that.
So, imagine the vesicle. Inside, they trap a chemical called ammonium sulfate. Outside, the vesicle is just floating in a normal buffer solution. Now, ammonium sulfate breaks down. The ammonia part, NH3, is a gas. It's tiny, and it can just slip right through the membrane and leave the vesicle.
Okay, so the gas leaks out. What's left behind?
The protons, H+,
Sure.
and they're stuck inside. This makes the inside of the vesicle very, very acidic.
Sexy.
Now, you introduce the antibiotic, Polymyxin B, to the outside solution. And because of that strong acid gradient, the PMB is chemically drawn across the membrane to try and neutralize the acid inside.
It's like a chemical vacuum cleaner.
It's effectively a vacuum, yes. The drug flows in,
Yeah.
and as soon as it hits that acidic environment, it becomes charged. It gets protonated, and once it's charged, it can't cross the membrane to get back out. It precipitates and gets locked inside the core.
That is just brilliant. It's a one-way door, a chemical trap.
It is. And there's one more beautiful little detail here. In nature, if you tried to put polymyxin B into a normal bacterial vesicle, the LPS on the surface would bind to the drug and just neutralize it.
Because PMB's whole job is to target LPS.
Precisely. It would disarm the weapon before it even got loaded. But because we washed our synthetic bacteria and got rid of most of the LPS, the antibiotic sits inside, totally free and active, ready to go.
So we've built the weapon. It's a hybrid lemon bacteria sphere, stabilized with cholesterol and chemically packed with a high dose of antibiotics. Now comes part 3, the journey. How do we get it from the vein to the lung?
We call it taxi, a living taxi, the neutrophil.
The first responders of the immune system.
They're the absolute foot soldiers. They just patrol the blood looking for trouble. So when the researchers inject these hybrid vesicles into the bloodstream, the neutrophils spot those outer membrane proteins we talked about. They see the PMPs, the pathogen-associated molecular patterns.
And the neutrophil thinks, aha, an invader.
And it does what neutrophils do. It eats it. Phagocytosis.
Yeah.
The paper has these amazing confocal microscopy images where you can literally see the vesicles sitting inside the cytoplasm of the neutrophil. They're not digested right away. They're just carried.
So the drug is now hiding inside the immune cell. It's a passenger in a car, but how does the driver know where to go?
The infection itself calls them. The bacteria in the lungs are damaging tissue, and that tissue releases these chemical distress signals called chemokines. This triggers a process called chemotaxis.
Right.
The neutrophils sense this chemical trail, and they just move toward the lung.
And this solves the barrier problem we started with. The drug on its own just bounces off the blood vessel wall. But the neutrophil, it has a key.
It has an all-access pass. The neutrophil performs this incredibly complex maneuver. It adheres to the vessel wall, it crawls along it, and then it transmigrates. It literally squeezes itself between the cells of the blood vessel and enters the lung tissue. It smuggles the drug right past the border patrol.
Okay, the taxi has arrived at the war zone. But the drug is still inside the taxi. Does the neutrophil just spit it out?
It's a little more dramatic than that. It essentially explodes.
It explodes.
It's a process called netosis. That stands for neutrophil extracellular traps. When a neutrophil runs into a super high concentration of bacteria or inflammatory signals, which is exactly what's happening in an infected lung, it activates a kind of suicide protocol.
Wow.
It ejects its own DNA out of the cell like a sticky, fibrous web.
Like Spider-Man.
Suicidal Spider-Man, yeah. This DNA web is designed to physically trap bacteria. But in our case, as that DNA web shoots out, it releases all the hybrid vesicles that were trapped inside its cytoplasm. It's like deploying the payload exactly at ground zero.
That is wild. It uses the body's own defense mechanism as the final trigger.
It's a perfect biological circuit. The immune system finds the drug, carries the drug, and then releases the drug only where it's needed most.
Okay, let's hit the 10-minute mark and lock this mechanism down before we get to the results, because that is a lot to process.
All right, let's do it. The sequence is...
First, the fusion. Lemon vesicles plus those gutted bacterial shells plus cholesterol makes a stable hybrid vehicle.
Then the loading. We use that pH gradient to suck the antibiotic inside and trap it.
Next, the taxi. Neutrophils eat the vesicles because they look like bacteria.
Then, the drive. The neutrophils follow the chemical distress signals right into the infected lungs.
And finally, the drop-off. The neutrophils explode in a process called netosis, shooting out DNA webs and dumping the antibiotic right on top of the superbug.
That's the whole journey.
On paper, that is incredibly elegant, but biology rarely works as cleanly as the diagram. So part 4. The evidence. Did it actually work? Did it clear the pneumonia?
They tested this extensively, first in vitro in the Petri dish and then in mouse models. In the dish, they put it up against CRKP, carbapenem-resistant pneumonia. The hybrid vesicle worked just as well as the free drug. And the scanning electron microscope images are just striking. You can see the bacteria treated with the hybrid have their membranes completely wrinkled and collapsed.
But the real test is the mouse. They gave mice severe pneumonia directly into the trachea.
Yes, and the results were statistically massive. They measured the bacterial load in the lungs, and the hybrid treatment reduced the bacterial count by 1.7 log.
And for listeners who don't speak log reduction, that is nearly a 99% reduction compared to the control group.
It's huge. And critically, it performs significantly better than the current standard clinical treatment, which is nebulized PMB plus IV Meropen.
Wait, it worked better than the cocktail doctors are actually using in hospitals right now.
Yes, and the mice live longer. In a separate sepsis model, where they injected the bacteria directly into the blood, the survival rate was 100% for the E. coli infections and 90% for the superbug, CRKP.
100% survival in a sepsis model that's almost unheard of. Usually you lose a few subjects just from the inflammatory shock.
Which brings us to the safety data. Remember the kidneys.
The whole reason we built this thing. Did it protect them?
They measured BUN and creatinine levels, the key markers for kidney function, totally normal. They checked liver enzymes, ALT and AST, normal. White blood cell counts, also normal. The encapsulation worked. It kept that toxic sledgehammer away from all the innocent organs.
I want to circle back to something you mentioned at the very beginning, the lemon. You said it acts as a medicine itself. The paper dives into the transcriptomics, the RNA analysis. What did they find happening at the genetic level?
This is the deep mechanism, and it's so cool. They sequenced the RNA of the immune cells, the macrophages, after they were treated with the vesicle. And they found that the treatment didn't just kill bacteria, it actively turned down the volume on the immune response.
It stopped the cytokine storm.
It stopped the cytokine storm. The RNA data showed clear inhibition of the NF-kappa B signaling pathway.
That's the master switch for inflammation, isn't it?
It's the big red button. It also inhibited the JAK-STAT and NAPK pathways. These are, the communication lines that tell cells to panic and flood the lungs with fluid. By blocking these, the lemon vesicles actually prevented the lungs from filling up.
So it's a true dual-action therapy. The antibiotic handles the bug, and the lemon handles the host's overreaction.
And the paper explicitly attributes this anti-inflammatory effect to the LNV component, the lemon part. It proves that using a natural plant carrier isn't just a gimmick. It has a real functional therapeutic role.
It is just fascinating that the solution to a super high-tech problem like antibiotic resistance involves something that's sitting in the produce aisle.
Well, combined with some very, very high-tech processing.
Fair point. Before we wrap up, I have to play devil's advocate. This whole thing relies on neutrophils acting as taxis. Is there a scenario where this fails?
That is the critical nuance. And the expert commentary on this kind of approach always points out one thing: neutropenia.
Patients who don't have enough white blood cells.
Exactly. I mean, who gets these severe superbug infections? It's often cancer patients on chemotherapy or people with really compromised immune systems. If a patient is neutropenic, they simply don't have enough neutrophils in their blood.
There are no taxis to hail?
Right. If there are no neutrophils to eat the vesicles, the drug just floats around in the blood. It's not going to get transported across the lung barrier efficiently. So this isn't a magic bullet for every single patient.
But for the majority of cases where the immune system is still active?
It could be a complete game changer.
That's a really important distinction. It works with the biology, so you need the biology to actually be there.
Correct.
Let's wrap this up with our final takeaways. What are the 3 things our listener really needs to lock in from this?
Okay. First, dual action. This isn't just a drug delivery system, it's a true combination therapy. You get the antibiotic to kill the bug and an anti-inflammatory to calm the patient's lungs.
Right.
Second, targeting. It solves the biggest problem in pneumonia treatment, getting the drug across the blood-air barrier by hitchhiking on neutrophils.
And third?
Third, safety. By stripping the toxins from the bacterial carrier and shielding the polymyxin B inside, it turns a dangerous last resort drug into a much safer, manageable treatment.
It upgrades a blunt instrument into a precision weapon.
And the implications go way beyond this one drug. This is a platform. You could load this with vancomycin or tigecycline, other drugs for other infections.
It really leaves us with a provocative thought. We spent the last century in this kind of chemical warfare against bacteria, just trying to find new molecules that kill them. But this research suggests the future might be more about hybrid engineering.
It creates a strange philosophical moment, doesn't it? We're wearing the skin of our enemy to save ourselves.
Which leads to our question for you, the listener. This whole method relies on tricking your own immune cells. Does the idea of injecting synthetic bacterial vesicles, basically fake bacteria, into your blood to cure an infection sound like a brilliant biohack to you? Or does it make you a little nervous?
I can tell you I'd sign that consent form in a heartbeat if I had superbug pneumonia, but I am curious what others think.
How would you feel seeing ingredients, E. coli shells and lemon juice on your IV bag? Let us know. Thanks for listening to The Deep Dive. We'll see you next time.